Welcome to Biology Made Easy. Biology Made Easy are biology lessons designed for high school and secondary school biology students. Students will find the lessons great that propels you for university. Welcome indeed. In our last lesson, we discussed biological drawings. You remember how to draw according to proportion, how to label, how your guidelines should go, etc., etc. We also discussed magnification, and at that time, we defined magnification of your drawing as length of drawing over length of object. Now, we want to look at something, all right? If I have this specimen, all right, and this is the length, this is the width or breadth. Now, if I draw this specimen like this, here is the length, isn't it? Now, what about if this is cut? And I draw the width. All right? So therefore, I draw the width as this. In this case, this becomes my length. So therefore, for biologists, mostly understand length, but magnification can also be defined as linear dimension of drawing over linear dimension of objects. So you talk about, whether you talk about the width, whether you talk about what is the linear dimension. So that is magnification of your drawing of large objects, macro objects we call it. Now, you can also observe that in drawing, you can describe the size. You can put on the size of the specimen. Well, let's look at this. This is an organism, a scorpion, drawn, and we have the scale as 1 centimeter. All right? It means that every 1 centimeter length, every 1 centimeter goes to the length of this organism. So looking at it roughly, every length like this is 1 centimeter. So another length measured 1 centimeter, another 1 centimeter, another 1 centimeter. You can get about 6 centimeters for this. So you can also put up a scale to show the size. And remember, magnification is also always written as times something, times 2, not 2 times. Magnification of your drawing is times 2, not 2 times. All right? Because we are going to look at something else today. We want to look at today's lesson. So what is microscopy? Microscopy is the science of the use of hand lenses and microscopes. So let's look at hand lenses then. Hand lenses come in various sizes. You can have some as big as this. You can have some like this. You can also have some as small as this. And the magnifying power of hand lenses are written on them. This hand lens, this one, had a magnifying power of 10 times. And this 10 times is written on the hand lens, the frame of the hand lens. So that is the hand lens. This is the frame or handle of the hand lens. And the magnifying power is normally written on the handle or the frame. Right. Now, we also want to explain that you take note that the hand lens, it's used in magnifying structures so that one can see very well. It's basically a convex lens supported in a frame. It helps us to see objects well, and we are talking about large objects, that is macro objects. So the hand lens allows us to see well. And please take note, if the hand lens is used to look at a structure so that you can draw well, when you have drawn the structure that you use the hand lens to look at, the magnification of the drawing has nothing to do with the magnification, the magnifying power of the hand lens. Now let's move on to look at optical microscopes. This is an example of an optical microscope. This is an optical microscope. We are going to look at optical microscope. This is a microscope slide. And this is a cover slip, very tiny. We'll have times to describe them. We are going to talk about these three pieces. The optical microscope, you see some in your shorts. There are various types of optical microscopes. Now, the optical microscope, it's used to see microorganisms. It's tiny organisms that your naked eye cannot see. For instance, this beaker contains water. And if the water has microorganisms, you cannot see them. So a little of the water on the slide, covered by a cover slip, and put under the microscope. This microscope is a device that will enable us to see the microorganisms on the slide. So what is the build of the microscope? The parts of the microscope we are going to look at. Once you understand the build of it, you will be able to see how to use it easily. And the optical microscope uses light. That is why it's called optical. It uses light for us to see the organisms. And it doesn't only use light, it uses also convex lenses. Now, this is the build of the optical microscope. Remember, we have a tiny organism on this slide that we want to see. We can't see with our naked eye. And this microscope is going to help us. So the microscope is simply, it has it has a structure which is called a stage. This is the structure that the microscope slide is put on. So if this is the microscope slide, it's a glass piece. So this glass piece is our microscope slide. And this microscope slide has our tiny organism lying on it. All right, that is the specimen. The green is a specimen. And this stage has a hole in it. So this whole stage, this is the stage, but it has a hole here. And our slide is lying here with the organism. All right, so let's have a structure that is holding the stage. So this structure is holding the stage in place. And we can't see this organism with our naked eye. So a lens, all right, is placed here. So this is a lens. So that this lens can help us to see this organism. This lens is not enough. So we have another lens up there. We have another lens here. And this lens is very close to the specimen. So we call it objective lens, if you like. And this lens, we are going to put our eye here to look at it. This is our eye. We are going to look at the specimen. And so this lens, very close to our eye, is called eyepiece lens, if you like. And this is our eye. But there's something that this structure will need to hold the objective lens. Another structure will have to hold the eyepiece. And this is the human eye looking through. So this human eye will have to look, is using these two lenses to look at this small specimen here. But then there should be enough light coming through, otherwise the specimen cannot be seen. So a mirror is placed here to trap light, all right? And that light is going to be trapped, so the mirror is also connected to this piece. And if light is trapped by the mirror, the light is passed through structures, two structures, to concentrate the light. The light is passed through two structures. One of these structures here is called condenser. And the other structure is called the diaphragm. All right, so basically, this is the setup of the microscope. Eyepiece, objective piece, all those are convex lenses. Then you have the stage. Then you have the diaphragm, condenser, mirror. That is basically the structure of the optical microscope. What is going to happen is that light is going to pass through the mirror, direct light, brings it into the condenser. The condenser concentrates the light, and then it passes through the diaphragm. You see that this diaphragm sort of is a device that increases or reduces the hole through which the light can pass through. So it regulates the light. Then the light goes through the hole in the stage. It goes through the microscope slide, then the specimen. So now the light is going through the objective lens, which enlarges the image of this object, and then goes to, the two are connected. These two are connected with a barrel. All right? So the light is goes through the objective lens. It magnifies the image. Then it passes into the eyepiece also, which is a convex lens that also magnifies the image so the eye can see. Finish. That's the optical microscope. So to be able to use the microscope, the first thing you need to do is to make sure the objective pieces are many. So make sure that the objective piece and the eyepiece, they are all in one line, so light is going through it. All right? Now we'll show you how you know whether the objective piece and the eyepiece are in line when you are operating it. We are going to look at how to use the microscope. Now let's get to the microscope. So, this is a very simple optical microscope. As we have described on the board, this just have a stand. All right? Good. It has a stand here, and we have the light inbuilt there. The simple microscopes you see in the shots, that you see a mirror there. This one has a light inbuilt in there. This mirror will trap light, all right, if it's a mechanical one. This is connected, there's a bulb in it, so there's light that will come in. Now, so this just directs light, all right? And then we talked about the condenser. Here, light from the mirror comes into the condenser. You see the shot, the diagram in your shot there has a condenser. All right? That was the older types of microscopes. But this one has the condenser built in. And so the condenser is this one. The condenser is here, under here. Has a condenser here. This structure here. And this condenser is also built in with a diaphragm. There's a diaphragm in there. You see that there are holes in there. So you can increase or decrease the amount of light getting through. So it has a condenser that concentrates the light from the mirror and the light and into the condenser. And this is a diaphragm. As we move this about, we are regulating both condenser and diaphragm. You see the old type microscopes? This is the mirror. In the old type microscopes, this is the mirror. It takes the light and puts it in a condenser. This is a screw that regulates the condenser. And then it comes into a diaphragm. This is a screw that regulates the diaphragm. Is that okay? All right. Then this is the stage. And our specimen will lie on the microscope slide on the stage. Let's use our microscope to focus, to look at our specimen on the microscope slide. First of all, you want to make sure that there's light in your microscope. Now, you need to bring the low power objective to be in line with the eyepiece. So you turn, this is the revolving nose piece that turns from one objective lens to another objective lens. So you move the revolving nose piece, all right, and turn, and when it's in line, you hear a click. So the objective lens and the eyepiece lens are in line, so there will be light in it. If you can't see light, you need to regulate the diaphragm and the condenser, so you have plenty of light. All right, now we put our specimen on the stage, hold it with the clips, make sure that the specimen is right in the center, all right, on the stage. Now you look with your eye in the eyepiece, look through and look at your specimen as you lower down with the coarse adjustment slightly. When you see the image focus, you can then use the fine adjustment to bring the image into a sharp focus. Right. So this is where we will end for today. You need to regulate using the coarse adjustment knob, this is it, to bring the image into focus. Then you use the fine adjustment knob to bring the image into sharp focus. Right. In our next lesson, we will continue from here to look at how you can use the high power objective lens to look at your specimens very well. We will also look at the magnifying power, magnification of the microscope. We will look at how to care for microscopes, and we will look at resolution of microscopes. So we meet soon. Bye-bye.